Hello everyone and welcome back to the channel. Today we'll learn an important and very practical topic from histology and cytology, lipid staining. Specifically, we'll focus on oil red O staining. Have you ever wondered how we can actually see fats inside cells under a microscope? In routine histology, lipids often disappear during processing. So how do we study them? That's exactly what we'll understand in this tutorial. Let us begin with the basic idea. Lipid staining is a specialized technique used to demonstrate intracellular lipids in tissue sections. One of the most commonly used stains for this purpose is oil red O, also known as ORO, useful for detecting neutral fats and fatty acids. Importantly, alcohol and clearing agents dissolve lipids during routine processing. If we used paraffin sections, the lipids would be gone before staining. The principle is simple. Oil red O is a fat-soluble lysochrome dye that partitions into neutral lipids, rendering them visible as red deposits under the microscope. Oil red O preferentially stains neutral triglycerides and some fatty acids. Polar lipids and phospholipids are less strongly stained by this dye. Because organic solvents remove lipids, oil red O protocols use frozen sections or avoid organic solvents to preserve intracellular fats for staining. A counterstain, typically hematoxylin, provides nuclear contrast so red lipid droplets are easily distinguished against a blue-purple background. Controls are essential. Include negative controls without lipid-containing samples and positive controls, such as liver or adipose tissue known to contain neutral fat. Always handle reagents and stains with appropriate safety measures, working in a ventilated area and following institutional chemical safety protocols. Collect tissue quickly to minimize degradation. For oil red O, preserve lipids by snap freezing in isopentane cooled on dry ice or by immediate freezing in liquid nitrogen. Store frozen tissues at -80 degrees Celsius for longer-term preservation. Avoid prolonged exposure to room temperature before sectioning. Section on a cryostat at 5 to 10 micrometers thickness. Thinner sections improve resolution but can be fragile, so handle slides carefully. Mount sections on clean glass slides warmed briefly to aid adhesion. Avoid use of organic solvents for fixation that would extract lipids. Fixation can be brief in 10% neutral buffered formalin or 4% paraformaldehyde for a short time, avoiding alcohol-based fixes that dissolve lipids. Rinse slides with running water and air dry slightly before staining. Prepare a filtered working solution of oil red O from the stock following recommended ratios. Pre-warm the oil red O working solution to room temperature or slightly warm. This improves dye solubility and staining consistency. Immerse slides in oil red O working solution for approximately 10 minutes at room temperature, monitoring staining intensity visually. Differentiate briefly in 60% isopropanol or rinse with distilled water depending on protocol to remove excess dye and reduce background. Counterstain nuclei with hematoxylin for a short period. Then rinse, taking care not to overstain which can obscure lipid signal. Mount slides using an aqueous mounting medium designed for frozen sections. Avoid organic mounting media that will dissolve the stained lipids. Label slides clearly with sample ID, date, and protocol used. Document stain batch and any deviations for reproducibility and quality control. Under light microscopy, oil red O positive lipids appear vivid red or orange red, while nuclei and cytoplasmic background remain counterstained blue or purple. For documentation, capture images at consistent magnification and illumination. Calibrate white balance and exposure for reliable comparisons between samples. Quantification can be performed using image analysis software to measure area or intensity of red pixels, normalized to total tissue area. Common pitfalls include lipid extraction by improper solvents, overstaining, and artifacts from frozen sectioning. Use controls and consistent technique to reduce errors. Always include positive and negative controls, record reagent lot numbers, and repeat staining if results are inconsistent for reliable interpretation. In summary, oil red O is a robust method to visualize neutral lipids in frozen tissue. Follow careful preparation, standardized staining, and proper imaging for best results. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.